which is all cells come from other cells. And I think although that's sort of obvious, it's also really important because in evolution, it operates, it's not just simply in development. So we're operating with a system which is hugely ancient, exquisitely beautiful, and I personally feel privileged to be able to spend time thinking about it. So I want to just keep emphasizing that point in the next image here. So in each of us, there are roughly 30 times 10 to the 12 cells. And in the adult human brain, there's almost 100 times 10 to the 9 cells. Now, we also now take for granted that we're all formed from a single cell, but this wasn't always known. And Carlo and I just had a bit of a discussion about this, but we can talk about this history in more detail. But I'll just assert here that the first clear statements of this concept were made by a Frenchman called Francois Raspail. And if you go to Paris, I encourage you to walk on the road that's named after him. And on the signposts on this road, he's named as a chemist and a politician. And I think the reason they call him a chemist is because there weren't really names for biologists when he was doing this. It was so early. And his political contributions were very interesting because he was actually politically very active. And he encouraged the French Republic, the French state, to invest in science. The French state was the first state to really, as a nation state, to invest in research, and Raspail played a very important role in that. Anyway, I think you'll get a thrill if you walk up the Boulevard Raspail. So in the adult human cortex, there are 15 times 10 to the 9 neurons. And we know that all of these cells are derived from a single cell. And that I say that the several hundred basic cell types of the adult are specified very early in development, when there are very few to low thousands of cells in the embryo. It's extremely important, this, because the patterning mechanisms that we're interested in only operate over very short distances. Now, interestingly enough, the brain consortium asserts that in the mouse brain, there are 5,300 cell types. Now, without getting into the sort of weeds of all of this, I think we all sort of basically know that the brain has the most complicated cellular structure of any organ in the body. And that's the topic of our interest together in this Zoom series. If you want to look at this discussion from the Brain Consortium, I provide a web link here that you can just simply click and you'll see their arguments. And in this presentation, I've labeled things that I want to encourage us to think about more, or that we've already discussed in the Zoom series with this blue star. I mean, I think Carlo and May and Soki have done a really great job in promoting us to set up a Zoom series like this. And I really appreciate their continued intellectual contributions to my thought. But I just want to say here, thanks very much, guys. It's terrific that you do this. The other thing that I've done, aside from putting a blue star in, is I've put some names inside a box like this of people whom we might invite in the future to talk. And in this first one, I've included Svante Pabo and Nenad Sestan and Chris Walsh, because I think all of these individuals would have very interesting things to say to us. I won't stop here to summarize what they might say, but I will comment as we go through the talk about a series of names that I've included in boxes like this. So in this presentation, I want to use it both as a way of reflecting on what we've already heard in the Zoom series, thinking about what future perspectives we might explore together, and who we might invite to speak. So my own interest in neurobiology started by asking a question which was prompted by this man here, Roger Sperry, who an American neuroscientist who first came up with what he called the chemo affinity hypothesis, where he proposed that neurons were chemically distinct from each other. And I went looking to see if Sperry was right, kind of uh, by using the hybridoma technology to probe for different neuron types. And the place where I really saw the most interesting um, result was when I used this tool to look at the nervous system of an invertebrate, the 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 leech, which is an annelid, and we came up with hundreds of antibodies, and so many that it really both showed that Sperry was right, led to the publication in Nature of a paper a long time ago now, but still the first really clear statement about the extent of the molecular biology of the nervous system. But for me, it raised the problem of how how on earth would you study all these different kinds of neurons? So I decided that what I was really interested in was the question of where do they all come from? Now, I want to introduce you to just briefly to the work of a very famous developmental biology group working in Germany before the Second World War. So their names are given here. Hans Spemann was one of the leaders of this group. Peter Newcomb and Johannes Holtfretter were his students. They each made very important contributions. I won't take the time now to spell out exactly what each of them did, but what was clear from their work was that the idea that there were cellular organizers acting on groups of equivalent cells, which they called equivalence groups, has inside it the notion of a stem cell. So, an experiment that was very influential for me was an experiment done by Peter Newcomb when he moved the notochord, which sits underneath the spinal cord, during the development of Xenopus, and showed that he could re-specify the position of motor neurons. So that the spinal cord, the neural tube, is an equivalence group. To use current terminology, it's a stem cell. So I went looking to see if I could find these stem cells, and in a series of experiments, found an antibody that recognized a very early group of cells before neurons are formed, and then cloned the gene that was responsible for this protein, which we called nestin, or neuroepithelial stem cell, and then found that there was a sequence in the second intron of this gene, which acted as an enhancer to drive the expression of LAC-Z in this case all over the developing mouse brain. So this very brief kind of summary allows one to say that neurons are diverse molecular machines derived from pattern stem cells. So this is just really to set the scene for us. And I'd like now to spend 1 or 2 minutes talking about enhancers. So there's been a real revolution in this field in the last 10 or 20 years. But what we know now is that the human genome contains hundreds of thousands of enhancers. And initially, when people started reporting this, it all seemed like too much to cope with. But modern tools, machine learning tools, and synthetic nucleic acid chemistry tools are really revolutionizing our ability to understand enhancers. And I star here three very recent papers. The top one is from Stein Erck's group. Many of you may have used the Scenic tool to look at some of your data. This is the same group here looking at the enhancer-driven cell type comparison in mammalian and bird pallium. The second reference is to John Rubinstein's group, and the third reference to a group that is at the Jackson Labs and at the Broad, and also has a prominent individual at Yale, Stephen Riley. And I'm encouraging us to think about inviting one or more of these groups to take part in our discussions. On the left-hand side here, I just show you something. Often we think of enhancers as being kind of almost like magical things. But here, even in this first definition of the nested enhancer, we show that the enhancer contains different transcription factor binding sites. And one of these, this red POW site here, which we label JKL, because these embryos here, JKL are shown with a knockout of that particular POW element, so that this is extremely important in driving expression throughout the developing mouse brain. So there's extremely interesting things that we could do now with the new tools that are available to us to use enhancers in improve our ability to understand neural development. And as we know, most of the genetic risk for psychiatric and neurological disorders actually is in non-coding regions. So our ability to understand these regions of the nervous system is increasingly of significance. As I say, I'm just trying to summarize things here to make it interesting, but any of these topics could be extended very usefully. And if you feel you have contributions to make to this, please interrupt or in one way or another contribute them. So in the stem cell field now, there's a tremendous amount of interest in making differentiated cell types to use clinically. And here, I just want to, again, to sort of phrase this in a historical context to show you the phases of certain individuals who've worked with me, who've played a very important role in the development of some of our ideas. This individual here on the top left-hand corner is Elena Cattaneo, who's in Milano. I could say wonderful things about Elena, but again, in the interest of time, what I'll say is that this reference here, Nature 1990, is one of the first times that people showed that you could make neurons in a dish from a precursor cell at all. That's why it was published in Nature, because previously there was this kind of belief somehow that neurons were sort of magical things that you couldn't really reproduce in cell culture. And next to her is Paul Teasar, who's done very important work on the nature of pluripotent stem cells, which we'll talk about in some depth. And below Eleanor and Paul are Vivian Tabar and Lorenz Studer, who are in New York. And I just want to spend a minute talking about some of their recent clinical work. So, because here recently, in the last week or two in Nature, are two clinical trials transplanting dopamine cells into Parkinson's patients. Spectacular progress, but over a very long period, right? So the first report that you could make dopamine neurons from pluripotent cells is this one here, Kim et al., Nature 2002. So over 20 years, it's taken us to reach the point where you can use human pluripotent cells to go into patients. And here we're still in phase 1 or phase 1/2 clinical trials. Now, I want to make a very specific point about these papers, which you can derive from the titles. So the top one here, a Japanese group, phase 1/2 trial of iPS cell-derived dopamine cells. And the second one, phase 1 trial of human ES cell-derived dopamine neurons. So in the Sloan Kettering group, with the involvement of a company and the investment of Boehringer, they're still using the human ES cell H9 in their clinical trial. Here, the Japanese group is using an iPS cell to derive their dopamine neurons, and it's an iPS cell that is histocompatibly matched with a large proportion of the Japanese population. Here I've put a link to a podcast where this is discussed very nicely. So I'll just leave it at that for now. But the point I want to emphasize is that we're still in the early stages of this process, and it raises the question of, do we have the best cell lines for transplantation? And by that, I'm not asking a quality control question here about that. That's why these people have spent so much time with these cell lines, is they've done all kinds of QC. But I'm asking a more kind of open-ended question about the variability between lines, and that whether or not we could, by using the variability that exists in the population, get better lines for Parkinson's disease transplantation and other therapeutic uses. So if you don't mind, I'm just going to keep going in this kind of fairly direct way, didactic way, just so we can get through this thing. But again, I just want to encourage you, if I'm not being clear, just interrupt and say to me that I'm not being clear. Now, if you want to get a better cell line for dopamine transplantation, it would be a good idea to know where the cell lines, where that particular cell, in this case, the ventral midbrain dopaminergic cell, comes from in development. And this cell comes from a very unusual place because remember, I just explained to you about Spearman and Holtfretter and Newcup talking about organizers and equivalence groups and how organizers act on equivalence groups to specify this differentiation of cells in the equivalence group. But Raja Katapa and others with us showed that dopamine neurons are themselves specified by the organizer, in the floor plate organizer of the ventral midbrain. So this is a very specific set of cells, and actually we still don't properly understand. There are many protocols for making dopamine neurons which work, but there's something about this property of being an organizer which still eludes us, and it's going to be a sort of the major topic that I want to raise with you, and that Carla will emphasize again with data at the end of this brief summary that I want to give. So here, our ability to make dopamine neurons was luckily for us, we started this off at a time where we were using mouse pluripotent cells, because the mouse makes dopamine neurons the mouse pluripotent cell makes dopamine neurons very readily. And many groups all over the world have repeated that. But when you turn to human pluripotent cells, the interest of variability is not so easily worked on when the dopamine system is used as your endpoint. And we focus then on the telencephalic phase. And that's what's shown here in a summary form. And this is where Nicola McKay was very influential. And it summarized all of his work with a single cartoon, and which is shown here, which is that Nicola showed that cell lines made from different individuals generated either rostroventral or dorsal-caudal telencephalic fates with very different efficiencies. And the reason they did that is they went through an intermediate state, which was the organizer state, either the hem identity, the dorsal posterior state, or the rostral patterning center identity, which gives rise to ventral fates. And this finding is now really central to our thinking about using pluripotent cells and organoids as a differentiation strategy to understand CNS disease. And I just want to emphasize here that briefly, using this reference here, which is from Michael Wigler's group, that in autism research, we've had tremendous advances, and a very startling observation it has been made by a number of groups, but this is a quote from this paper, that in ASD, de novo mutations contribute 50 or more than 50% of cases to 50% of cases in low-risk families and 30 to 40% of all autism cases. And this is really stunning statement, de novo mutations, because de novo mutations means that the mutation is affecting the lineage in that person, and it's affecting the lineage in the formation of the brain. So many of us kind of know this, but I just want to emphasize it here, how remarkable this is. And one of the reasons I think it would be a lot of fun to invite Chris Walsh to talk to us is that his group, I mean, there's other groups we could usefully talk to as well, but his group has done some very important work here. And I mean, Flora may still be on the call, but Flora's group's also been heavily involved in this kind of work. And Flora's group is the source of this next reference here. Alex Jordan may be on the call. This is a very influential paper showing that in autism cases, using forebrain organoids, you can actually map out functionally different behaviors for different risk populations. So We've also heard from this group here, genetic variants associated with longitudinal changes in brain structure. In a very recent um, uh, Zoom together, we heard from Jason Stein. And and here, finally, I reference another paper that's just coming out in Nature Communications from Nicola and his colleagues, looking at the distribution of risk in the early stem cell component of the nervous system. So again, forgive me for being very brief in the way I'm summarizing this, but the main point I want to raise here is that it's absolutely unequivocal that the neural stem cell compartment is a source of risk for psychiatric and neurological disorders. And if you're interested in this, you have to understand the behavior of these completely non-linear cellular states, which we're calling organizers. So when we saw that kind of result in which appeared in our culture system around 1 week or 2 weeks of differentiation, we naturally started asking whether the cells are different from the very beginning, from the pluripotent state. As you grow them, are they different? And as they first enter the neural state, do the cells behave in different ways? And here, this is work where Sul-Ki Kim and Sung-Mei So, who very likely on the call are were central players. And in a very simple assay system, we could show that cell lines behave very differently. So give me here, but just I want to just draw your attention to panel A. Panel A is really easy. You're just growing the cells for a week in culture. You passage them, you treat them with a ROCK inhibitor, you take it away, you call that day 0. You grow the cells for 6 days in 3 different conditions: self-renewal, NSB to promote neurectoderm differentiation, BMP4 to promote mesendoderm differentiation. And already you can see these 6 cell lines that we focused on initially have very different behaviors. This is NANOG. We'll talk about NANOG as a marker for the formative or early naive pluripotent state. SOX21 as a marker for the very first events in neural differentiation. Now, I think the first talk we had was from Sally Temple's group, which is referenced here. And Taylor Bertucci showed that you could actually standardize his cell lines by the appropriate application of the NSB inhibitors in the system. I think that's extremely interesting. Here we haven't done that. Well, there's a very interesting discussion to be had here about can you really standardize this system and what does it mean? It's also true that although this is very simple, there's a huge expansion of our knowledge about far more complicated differentiation strategies. I've cited Sergio Pasca and Jun Yu here as individuals whose labs have contributed greatly to our understanding of complex systems in pluripotency. Sergio is well known for his work on organoids and assembloids. And here, this is the work from Liberali's group in Switzerland, where they did a very detailed analysis of gastroloid development. Again, I'm not going to emphasize here the details of this work, simply to say that there's a lot of interesting issues here, and we might well want to invite one of these individuals to come and talk to us and think about it together. Let me just keep moving forward, though, by showing you that if you take these conditions and take this poly-A plus RNA-seq, bulk RNA-seq, deep RNA-seq, ribose 0 for the most part, here you can look at the distribution of this RNA-seq data in a simple PC1, PC2 map, and you can make some very interesting observations. So black here is the self-renewal condition. Red is the early neurectodermal differentiation, and blue is the first step in mesendodermal differentiation of these cells. We're going to look at this quite a bit together in the next 15 minutes. So just sort of do what Sidney Brenner called a neurox of this image, right? There's really detailed information here. The black is all the cells are more or less on top of each other in the black. But when they start differentiating, you can see the different cell lines start behaving quite differently. And one of them, this one here, SA1, is differentiating the furthest in this NSB condition, consistent with this large expression of SOX21 over here in this core domain of this epithelium. And this line here, I4, is differentiating the furthest along the BMP pathway, consistent with it being at the opposite extreme of this continuum here of cell line behaviors as you map them out with NANOG and SOX21. We then did an analysis of this gene expression data using a tool which is called COGAPS. COGAPS is an agnostic, non-negative matrix factorization, which simply assembles your data set according to the patterns it sees. And I want to show you the two major kinds of pattern that this analytical tool shows you. It shows you some patterns that are specific for certain cell lines, shown here in B. Every cell line had its own pattern. These patterns are very stable, and they occur both in vitro and in vivo. So for some of these individuals in our work, we had the forebrain had been sequenced as well as having pluripotent cells from that same individual. We found the same pattern. Excuse me. I have to restrict myself from waving my hands around too much. We found the same pattern in the pluripotent cells from that individual as we found in the adult forebrain of that individual. Forgive me for being kind of colloquial with you for a second. That's cool. What does that mean? It means we're all different. Does that matter? Well, one thing that we see in these cell identity patterns is we see that these cell identity gene expression patterns involve highly specific sets of genes, and particularly more recently evolved genes. Here I cite names of people who can speak to these issues in interesting ways that we might invite to talk to us if we want to think about these problems more. Here I want to show you an identity pattern for a particular cell line. Here's the particular pattern of OTX1 in all of these samples. Red is the neurectoderm differentiation. Blue is the signature here is very low for blue in most of the cell lines. But one of the cell lines shows an enhanced signature in blue and an enhanced signature in black in self-renewal, as well as this enhanced signature in red. And the COGAPS pattern splits this into two different patterns. One is this pattern 12 here, which we'll talk about more in the next slide, but which is a neurectoderm specific pattern. And one is a cell line identity pattern. So in all of the conditions, this cell, UCO6, is enhanced for OTX1 expression. This is an example of a gene which has a strong cell identity component. And over here in C, I'm just showing you that these cell identity signatures, in this case, the H9 signature, was found in many different data sets generated in our own lab and in other labs. These are very stable features of these cells. As we've said, H9 is the cell line that's being used in the New York Blue Rock Therapeutics clinical trial. In addition to these cell identity signatures, we found dynamic signatures shown here, which vary according to the conditions that we employed or the time. Here you can see for OCT4, the OCT4 pattern is this. This is the pattern of gene expression here. This is RPKM that's being measured. But the COGAPS analysis splits this into two different kinds of pattern. One is a pattern which is declining steadily over time in the self-renewal, slightly more abruptly in the neurectodermal differentiation, most abruptly in the SAO1 cell, which is differentiating faster in this condition. And then if you treat these cells with BMP, this is turned off very fast, but most fast in I4, which is already moving forward along that trajectory faster, even in pluripotency. So I just want to make a comment here with these papers here. There's a huge advance in DNA sequencing. So we now have long reads, telomere to telomere reads of increasing numbers of human beings. You will see these cell identity patterns in these data sets. So I just want to emphasize that that might be a topic that we really want to think about together. If you're really interested in the individual identity of particular cell lines, access to this kind of DNA sequencing data is said to predict the transcriptome. So that's a huge advance if that's true. Let me keep going with a focus on these neural patterns and, and just show you that there's two kinds of neural pattern that appear in this analysis of these data. One is a pattern called P15, which we show in this upper panel here, and one is a pattern called P12, which is in the lower pattern. P15 shows you, as soon as you start differentiating these cells in NSB, almost all the cells start showing an increase in the expression of a particular set of genes, the set of genes that COGAPS identifies as pattern 15 genes. SEO1 again shows the clearest movement along the space, and then a decline. Pattern 12 occurs later, and again, SEO1 shows the clearest move forward, and it's a trajectory which takes us into forebrain development. As you can see here, when we take these patterns and project them into mouse gastrula RNA-seq data that comes at this stage from John Marioni's lab while he was still in Cambridge. Oh, I see I've got John Marioni's name down twice here. That's not an accident, perhaps. John Marioni is now in Genentech, and John Marioni we might really want to invite. All the data that Roche are generating across the different Roche sites in the world is going through John Marioni's group in the Bay Area in San Francisco. So this is hugely interesting. But this image here of the developing mouse gastrula is the second image I'd like you to neurox. This is an extremely important study in my view, because it gives you very beautiful insight into the development of different tissues. Here in the mouse, look at the way pattern 15 is spread out across this space here. And in contrast, pattern 12 is moved slightly further to the right. Here, if you look at the annotation from Marioni's group, Pijuan Sala et al., this space here is called rostral neurectoderm, followed here by what they call spinal cord, forebrain, midbrain, and hindbrain. And then above it, spinal cord. And at the end here, this sort of peninsula right at the end is neural crest. These images are really worth sort of just incorporating into your frontal cortex for a minute or two, into your working memory, because we're going to talk about them 2 or 3 times in the next 15 minutes. But this confirms that in vivo, that these patterns that we see in the pluripotent cells indeed represent an earlier and a slightly later stage in neural development. I'll just show you here. This is the expression pattern of SOX21 itself in these different conditions and these different cells. It's these data that the COGAPS morphs into P15. We could talk about it more. You can see that there's actually an identity component. This circle, this black circle here, says that SCO1 already has high levels of SOX21. So if you're really into this, you'll be thinking, oh, wow, that's cool. Yeah, it is cool. And this is the SOX21 image in the gastrulating mouse embryo, as opposed to the pattern 15 image. So let me just keep going. So we have done a lot of work on this. We've knocked out SOX21. We know that SOX21 is doing something very interesting. It's a repressor. It's a member of a group of genes along with SOX1, SOX2, and SOX3, which are activators in what's called the class B SOX family. SOX21 is a repressor. And here what we showed is that it represses, in particular, the expression of genes that are involved in early BMP-induced induction. So that's the way it promotes forebrain differentiation. It's because it's repressing the BMP system. I'll, I'll now switch to a second set of experiments that we did on these cell lines, which show that they differ in their retinoic acid responses. So as I've explained to you, each of these cell lines has an identity signature. Here in red is the identity signature for SAO1 mapped out against the overall expression of gene values, gene expression values in SAO1. So the SAO1 gene expression specific signature is mapped in red, and it maps very closely with with the with the overall pattern of gene expression in these sample gene amplitudes. In I4, though, we saw a very interesting thing, which is that the I4 signature shows this real bump here. And this bump is what's mapped in red here in the I4 identity signature is the retinoic acid response genes. And in particular here, there's a gene called GBX2, which was well known in developmental biology to be involved in specifying the forebrain, the midbrain forebrain boundary in brain development. So here it looks like retinoic acid response genes are a major part of this component of the I4 signature. They're highly enriched in the I4 identity signature. And here you can see that when you treat these cells, all these different cell lines with the retinoic acid, the different cell lines labeled here with different symbols and different colors, so different expression of HOXB1, a posterior marker, in response to this retinoic acid dose response curve. And here you can see that when you map out the retinoic acid response gene enrichment in their identities against the induction of HOXB1, you can see it's almost a straight line, that the retinoic acid response here signature predicts the differentiation of hindbrain fates. And here we can link this to the SOX21 result I was telling you about, where either pharmacologically, in this panel here in C, with the first panel in C, our retinoic acid receptor beta inhibitor turns on SOX21 specifically in I4. And here, with a SOX21 knockout, you can see that as opposed to the wild type, which is in red, the knockout of SOX21 leads to enhanced enhanced HOXB1 expression. So that allows us to draw an image like this, which is that the forebrain is driven by SOX21 by its action as an inhibitor of BMP-mediated mesendodermal fates, and the hindbrain is promoted by a retinoic acid response, and particularly by the retinoic acid receptor beta. And this deserves a red star as being a clear example of the action of an organizer in a very simple system. Now, we've done a lot of work on this that I'm just skipping through. We've done this on other cell lines. This seems to be a general feature of cells of human pluripotent cells. But the simplest way for me to show that is to show it here. So here we're using data that was generated by Ihor Lemishka's lab many years ago now, where they RNA sequence of 100 different people, pluripotent cells made from 100 different people, which they had made in replicates. So there's 2, 3, and sometimes 4 replicates of any given individual in this pluripotent system. And here we're just plotting out the PC1 of this RNA-seq data. That's just shown here as this curve. This is high PC1. And then the individual's cell lines are colored by the expression of the retinoic acid receptor gene level, this signature. And you can see that the high PC1 shows high levels of retinoic acid receptor gene expression. What that tells you is that many tens of individuals in this 100 genome sample show elevated retinoic acid receptor gene expression, we would infer that they would show biased hindbrain differentiation. It's not just a specific feature of one or two cell lines that we happen to study. This was tremendously influential for us. Now, there's all kinds of details here that are interesting, but overall, I just want to take you back here to this kind of mouse gastrula map that comes from Marioni's lab from the Pijuan Sala et al. Data. If you take this high and low PC1, this PC1 distribution, and map it into the mouse, what you see is that high PC1 maps to very specific regions here. To this lower region here, which is the endoderm, and this upper region here, which is components of the mesoderm, whereas the low PC1, shown here on this heat map, maps predominantly in this, right on this other side, which is where the earliest cells of the mouse gastrula exist. And particularly here, I'm going to show you it's really on the left-hand side of this that it maps. And then to some extent in this region here, which is the region we've already identified as being the forebrain expressing regions, which are elevated in pattern 15 and pattern 12. Tremendously cool, right? So there's this notion, for example, that all pluripotent cells are by default biased towards brain development. This result seems to contradict that and suggest that large numbers of pluripotent cells are actually biased towards hindbrain fates or biased towards mesendodermal fates. Now, I've just made a note here about stem cell banks. So here we're using this work from Ihor Lemishka's group, but actually there's many cell banks that are available to now, and many of you on the call may have banks that are available to you that you've generated. So this deserves a much more detailed discussion than I'm giving it here, and it's of great interest to think together about how we might employ other data sets to explore these kinds of ideas. So now we're really coming down the pike here. Now I want to sort of shift gears from talking about work which is published and you've probably heard before, and now start emphasizing what it means, why currently I'm so interested in this and why I'm spending time introducing you to it. So here I showed you PC1 of these Lemishka 100 genomes. But here, let's start over here at C. What we're mapping here is PC1 on this axis and PC2 on this axis. It's just a really simple way of distributing the cells that we're looking at. They're again colored by retinoic acid gene expression level in the Lemishka data. And what you can see is that there's a very specific shape to this, that the retinoic acid receptor high samples are all down in this lower sort of left-hand corner, and it's this upper right-hand corner which shows very low expression of this. So what does this mean, actually, Ron? So if you take data made from the developing mouse embryo, here this is commonly referred to as a corn plot, and these individuals here, this is Patrick Tam's group from Australia, cut sections of the mouse egg cylinder, and they sequenced samples where they had transverse section, which then they sectioned further into 4 domains: anterior, posterior, and left and right. I'm just showing you one example here. PC3 of their data maps like this in the mouse, very high anterior expression in the developing mouse embryo. And when you project these data into the Lemishka data set, it maps here. This is anterior. This is maps like mouse anterior. This and this image show profound similarities, informative similarities. This shows a bias towards brain, and this shows a bias towards primitive street and mesendoderm. If you do a similar thing here, but you take the macaque gastrula and project it into the same data set, here you can see it's colored by different cell types in the macaque gastrula. The epiblast cells map here in gray. The macaque brain cells map here. The nascent mesoderm cells map here. And the endoderm cells map here. There's a map here of all regions of the macaque gastrula in the pluripotency space, which is why I write here, these data define a complete map of developmental variation across the human population. You'll forgive me perhaps for just emphasizing this to you. I think very often when people talk about variation between cell lines, we're sort of left with the feeling that people kind of switch their brains off. They think this variation is too complicated to understand, but actually it's not. There's a map of the early developing embryo in pluripotency space. Our task is to find out what this map means. And what I've suggested to you already is that one thing it means is the cells have different developmental properties. So let's keep exploring this together for 2 or 3 more slides. And again, I just really want to emphasize, at a time when the scientific community is being criticized for being irrelevant, I just want to emphasize, it seems to me it's our obligation to say why we think our work matters. In this next image here, we're using data generated by Kelly Fraser's group in San Diego. And if you recall, Timothy Arthur talked to us about this work. They took over 200 human genomes and did RNA-seq, and they used a number of informatic tools to generate gene networks, regulatory neurons, regulatory modules, gene regulatory modules, which they associated with different behaviors in the cell. One was called formative, and this one I've called neural here is the one that they called more primed. You can see why I'm calling neural and primed very similar as we go through this. If you take their networks, GNM10 and GNM5, and project them into our Lemishka PC1, PC2U maps, you can see that the formative network maps over here in this space in the right-hand bottom corner, and the neural primed network maps over here in this on this left-hand side of this space. We could spend time looking at the other networks. They have over a dozen networks defined in their paper. But what I want to emphasize here for our purposes is what this means. So in this GNM10 network, they have Wnt4. And in addition, they have SOX21 and 10, SOX10, SOX9, OTX2. Over here is Wnt4 and a series of frizzled genes. In the formative network, they have Wnt3 up here, Mix-like-1, OCT4, lefty, nodal, matrix metalloproteases. Let's just focus on the 2 Wints. As we know, and for example, Jason Stein was talking about this, but there's a lot of evidence that shows there's Wnt mutations that are autism risk mutations. Wnt3 is in this formative network here. How does Wnt3 itself look when we look in the pluripotency UMAP? Wnt3 is distributed in this space. This is consistent with this idea that this is formative. When we take early human pluripotent cells that I've introduced you to, H9, I4, and SA1, in short times after passage, or differentiating down to telencephalic fades, or differentiating to mesendodermal fades, you can see that Wnt3 is somewhat expressed after you passage the cells. That's interesting if you're really interested in the formative state and the levels of expression of Wnt3 that are consistent with signaling in the formative state. But in the early telencephalic state, Wnt3 is hardly expressed at all. But in contrast, when treated with BMP, within the first 24 hours, is turned up in all 3 of these cell lines. So the reason Wnt3 is in this formative system is not simply because it happens to be there and sort of abstractly Wints are important. It's there because this Wnt3 is involved in regulating the differentiation of cells out of this formative precursor state into the later mesendodermal state. And in contrast, Wnt4 is on this side of the of the pluripotency UMAP. It isn't induced by BMP. It's expressed at very low levels in the formative state, but the cell line, it is expressed in its SAO1. But as soon as you start differentiating the cell to NSP, up it comes. Now, I'm just going to go slowly for a minute here. For me, this is one of the most important images that I'm showing you, because it's taken me a long time struggling to understand how are we going to make progress on this kind of topic about the signaling pathways that are present early on that are controlling bias. And I think here we're beginning to see how this is going to play out. I hope that's good enough to persuade you that at least this is interesting. Let me go on and show you another image from pluripotency, which I find very impressive. Here's the gene FESF2. If you go and ask people in developmental neurobiology what FESF2 does, they'll tell you it controls the differentiation of the forebrain. And the reason they'll tell you that is because if you knock it out in the mouse doesn't have a forebrain. It doesn't have a forebrain. It's missing. Here, FESF2 is expressed in a very specific group of pluripotent cell lines here, in this region of the UMAP. If you pull out just the PC1 data of Lemishka's work and look at the expression level of FESF2 in all of these individuals, you can see on this left-hand side, there are a number of individuals who show high expressions of FESF2. This is RPKM we're measuring here. We know the level of expression of FESF2 when we differentiate cells to forebrain. This is the equivalent level. These cells are already expressing FESF2 at very high levels. And these individuals over here are flat. They're not. Here's an image from the mouse gastrula, which shows you where FESF2 is on embryonic day 8.5. It's right here on this edge. That's the forebrain developing. Well, you ask yourself, is this the first time you see FESF2 in the development of human embryos? And the answer to that is really quite startling. This comes from the work here, referenced Nature Methods 2024. It's from Josh Brickman's group in Copenhagen. This is the oocyte, zygote, pronucleus, zygote, 2-cell, 4-cell, 8-cell, late morula, trophectoderm, inner cell mass, epiblast, primitive endoderm. This one here is unknown. You can see FESF2 is already expressed at relatively high levels right at the beginning. Remember, human zygote to genome activation is said to occur at day 8. So this is either a very unusual gene which is transcribed at this early time, or these are transcripts that are inherited from the oocyte. There is expression in the oocyte, but the expression appears to go up. Yeah, I just used Bambi here, a BMP responding gene, to show you a contrast where there's no expression at these early times, and then it's clearly turned up. So there's a lot of interesting things that we could say further about this, but I just want to emphasize the importance of this by saying it's not just FESF2 that shows you this early expression. I'll just give you 2 other examples that show early expression like this, OTX2 and EMX1. So it appears as if there's a bias in these very early cell types towards very specific anterior neural fates. That's surely going to be interesting. This work here is in human. The Brickman data also does a very careful analysis of comparing human and mouse early embryos. Human and mouse early embryos are not the same. Their work shows that, and so too does this, I think, very important paper from a group at the Crick Institute in London. And here I'm referencing a work from Joe Gleason's lab, which suggests, along with several other studies, that there's mosaicism in the sperm that's affecting risk for psychiatric disorders. So the role of these very early cell types of the embryo and the gametes in regulating the early development of the brain is beginning to open up for us. Very cool. These early events appear to matter, and very early events also appear to matter. So I want to show you this image now. Here, you've seen the gastromat before. I don't really need to go through what it is. This is work from Marioni's group. Vishwan Sala is not the first author here, but I think the second author. And what they're mapping out here is the differentiation towards somites in the mouse embryo. And they're showing that the origin of the somites appears to come from more than one developmental pathway. And I think that's something that we might bear in mind here. We tend initially to be dominated by our views that there are single pathways differentiating cells to specific fates. But actually, as we look in more detail, it's becoming clear that there's actually multiple pathways, and that's extremely interesting. The reason I want to show you this, though, is because one of the important cell types that's generated by these pathways are motor neurons. Motor neurons are clinically interesting in the case of ALS, but they're also a very interesting developmental model. And what this work suggests is that these NMP cells, which are understood in some depth, particularly by the work of James Briscoe's group, start off here. These are the NMP ancestors. You notice here, there's a border across this very early pluripotency space. And these cells are mapping out here across this differentiation trajectory. This border is extremely interesting, right? Here's FGF8, a patterning gene that we've already thought about together. And here you can see the border in the early space. And then when you look 2 days later, and when I was showing you these images earlier, Silky, you asked me, why did I call this 2 days later? Why did I call it human system 2 days later? Here you see in the mouse, at embryonic day 6.5, these are all the cells in the mouse embryo, dominated by this pluripotency space. Epiblast here and primitive streak here. Extra-embryonic ectoderm here. Anterior visceral endoderm right at the tip of this primitive endoderm component. This anterior visceral endoderm, of course, for developmental biologists is hugely interesting because the anterior visceral endoderm contains the initial patterning center that specifies the formation of the forebrain. Okay, that's Rosa Beddington. So here you can see FGF8 starts off here in the primitive streak, but when you look 2 days later, in addition to being here in posterior somites, here in posterior somites, right, here in posterior somites, here in posterior somites, it's also in the hindbrain. And perhaps also here, yeah, this gets very interesting. This little tip here is actually the floor plate. So it's in posterior neural regions. This work here, Zhang et al., Nature Neuroscience, is a detailed discussion of why motor neuron formation is extended in the human system. And I won't stop now to really go into it with you, but it's really beautiful. And it's a very detailed analysis in terms of NKX 2.2 and OLIG2 of how this works and how it's different in the mouse and the human. The next paper is from Lorenz Tudor's lab, Nature, last year, talking about what controls the timing of differentiation of human neuronal maturation. These two papers here, one's from one's from David Hauster's group, and another from Van der Hagen's group, talking about how recent mutations in this particular notch gene are regulating neurogenesis in human cortex. These individuals here, Madeline Lancaster, Alex Pollan, and Derek van der Kooi, all would have very interesting things to tell us about these questions that I'm just sort of introducing you to here. One more data image that requires your frontal cortex. Here's an image that comes from a Nicola paper in 2023, where they dissected different regions of the macaque frontal cortex, telencephalon, and defined all of these different cell types, patterning centers. It's fabulous, right? Remember it right at the beginning of this, which is, yeah, I'm taking too long, but let me just finish. I was talking about the brain consortium and how they said there were 5,000 cell types in the mouse brain. So you shouldn't be too surprised to see that in the macaque brain, there are all these different cell types defined by Nicola's single cell RNA sequence data. What's really interesting about this map here that he shows you is that although initially you look at it and you think, wow, wrong, here's another really incredibly complicated map that you're expecting me to understand. Actually, it's not so complicated. Here's our spondin 3 in patterning centers in some of the uh, samples that he calls dorsal neural stem cells. Here's FGF17 only in this sample here from patterning centers. Here's SOX21, slight expression in that sample, in another sample from a patterning center. And then expression here in this group of cells, dorsal neural stem cells. And then here's eomesodermin, which most of us know if we're paying attention to cortical maps, is expressed at a very specific time, just before neurogenesis in the cortex. So here you see a transition from a SOX21 neural stem cell into an eomesodermin state, and subsequently you're going to get excitatory neurons appearing in the system. So you can understand these maps when you spend a little time thinking about them. And as you go through this, here are the ventral domains. Here's inhibitory neurons. There's lots of things to say about this. Here's microglia. Here's astrocytes. In his paper, Nicola talks about these patterns in some detail, about how they appear and disappear, how disease risk maps into this space. It's all very cool. What I want to show you, though, is if you take just 3 examples of genes that we've thought about in terms of patterning space. Here's one we've talked about in some length, FGF8. You know what to make of this. Here's FGF17. Here's eomesodermin. And here's our spondin 3. So all of these patterning genes are already at work in this pluripotent state. And I'm just going to argue that these papers here, for example, the recent paper from Flora's group, would be very interesting to discuss together in some depth for us. I hope that's a good enough example. The point that I particularly want to stress to you is it's my view that we're going to understand the very early stages are going to give us very powerful insight into how these systems work. I'll finish by showing you this image. You've already seen these kinds of images. Here we're projecting human next-gen data. Here, the human erythroid data maps here, human mesokine maps here, human endothelium maps here, neural tube 1, 2, and 3, forebrain, midbrain, hindbrain, spinal cord. It's our view at the moment that this bottom left-hand domain has not yet segregated in a very precise way into these different states. But if you remember, FESF2 mapped in a very specific set of cells here. So there's a really important details to be obtained by looking specifically at the behavior of different lines here. But the overall point that we're making is developmental potential varies in the human population at pluripotency. To know more, as I keep saying, about how these lineage cascades play out. But we've also had a talk from this group here at the Allen Brain Institute in Seattle. And remember, this is what the title of their paper, Inter-Individual Variation in Human Cortical Cell Type Abundance and Expression. So what they're saying is that their analysis of RNA-seq of their human brain samples suggests that different humans have different numbers of cortical cell types. What would be a really good way of getting different numbers of cortical cell types? It would be really good if these lineage mechanisms didn't work the same in every human being, but they gave you different numbers of cells to start with. Summary, same statement above. Remind you of the distribution of SOX21 and RAR beta, both in the pluripotent cells and at the embryonic day 8.5 mouse gastrula. You can already see the forebrains at this tip. Remember, this is where FESF2 was. Hindbrain mapped out by RAR beta. these two systems oppose each other. That's the point. So thank you for listening to this. I hope you find it interesting.
Thanks,
As I
Ron.
say, it's something that we'll all have to contribute to. I've been trying to talk in a clear and as rapidly as possible to keep the ideas sort of simple and coherent. Hope I succeeded to some extent, anyway. Okay, thanks very much, guys.
So I was going to jump into some views of data to almost summarize what Ron was talking about, but jump in with questions as we start to move to that. That'll just be a minute or two here at the end. And my goal here is twofold. One is to again reinforce that the everybody we now, we're becoming increasingly aware that variation, both genetically and epigenetically driven variation between humans, is accessible and quite clear in the pluripotent state. And that it has impacts not only on what their cells will do in a dish as we try to push them through different lineage differentiation protocols, but as Ron mentioned in that neuron counting paper, that it has impact on how we build our bodies. So that's the first thing I want to emphasize here at the end. But the second thing I want to emphasize is data and systems that we can use together to interact on these ideas that can push ideas forward and extend our understanding of what's going on by exploring data together. So this is a different visualization of some of the data Ron was going over. This is the pluripotent stem cell data on Nemo Analytics. It's a public website. I've sent many links around, and I want to encourage us all to use it more, or any other data-driven methods to interact on these ideas. But what this first panel is the same data Ron had been showing us with the 6 lines that show different behavior in pluripotency, NSB for neuroectodermal differentiation, and BMP4 for mesendodermal differentiation. And he had mentioned pattern 15, this sort of first step in the emergence of the neural lineage from pluripotency, and that it was very different from cell line to cell line. So what this Nemo Analytics website is good at doing has 2 big pieces that together are super versatile and super powerful. One is getting data together. So we've taken a lot of time to put interesting data sets together so you can look at them. And then the second thing is what you can look at in those data sets. The first is simply genes, but here what we're looking at is a gene signature. One of the 22 patterns, in this case, pattern 15, from the patterns that were generated from that COGAPS NMF that Ron mentioned. So just to remind us, what was this pattern? In pluripotency, it's quite low, but maybe a little bit of action here. Once we hit the cells with the dual SMAD inhibition, SAO1 leads the way, but actually wraps up this pattern. By day 6, this one's going down, and Ron also showed us the next pattern, pattern 12, which is the sort of second step in the emergence of the neural lineage. But again, the point here is that the different cell lines are all starting to go down this path, but with differing speeds and efficiencies. And we can see that here. Okay, so I'm going to move a little bit more, but in each of these cases, remember that we can look at this pattern 15 across a huge number of data sets. I'll show you only one more here so that you can remember. This is the Pijuan Sala data from John Marioni's lab. And again, this is a projection Ron already showed you. This is the developing brain in the mouse gastrula, where pattern 15 shows up. Turns out it also shows up a little bit in the notochord. Now, there's pictures all over here, and I don't want to get too complicated. So I'll just leave you with this one where we see pattern 15 in our pluripotent stem cells. I'm going to go to a different page now where we've loaded up Tim Arthur's gene network modules from the paper with Martin Pera and Kelly Frazier. And as Ron mentioned, their module 10 intersected with our observation on this neural component. And you can see here, this is almost the same as our pattern 15, with SAO1 leading the way, and by day 6 shutting down this module. But what you can also notice here is that just by looking at the colors, not even worrying about what is a cell line, that this module that they called primed pluripotency, that Ron was referring to as neural element of pluripotency, if you look at the pluripotency data in our hands of these 6 lines, this light green is one cell line, the red is one cell line. Each of these cell lines has sort of a different idling rate for the power of the neural lineage within pluripotency. And what we and others have now shown, as Rod went over, is that that difference within pluripotency is a profound predictor of those cells' behaviors when you do differentiate them. And I think it's sort of a grand challenge for the field right now, and Ron, you mentioned the same thing, is can we bridge the gap? Now, the gap now is between how does someone get 10% more of some kind of pyramidal neuron in their brain, and in those people, we might be able to perceive or study something that's different about their stem cells. So really that is what I see as the grand challenge in pluripotent stem cell biology. Can we start to
Let me interrupt you, Carlo, just
Yes,
for a
jump
second,
in,
just to
please.
emphasize that point. So, in Parkinson's disease, it seems like one of the biggest risk factors for Parkinson's disease is that different individuals have different numbers of dopamine neurons right at the beginning of life. And that we're all losing dopamine neurons, but if you start off with lower numbers, you hit that threshold sooner. So I'm just giving it as an example of a situation which really matters to human beings, the answer to this question that you're raising.
Yes, I remember your study of FOXA2, where you knocked it out one copy in the mouse to show exactly what you were talking about. And the analog there would be perhaps a common
Yes.
variant in a FOXA2 enhancer that could
Right.
influence the generation of neurons at very early stages. But then, as you say, the phenotype you wouldn't see until you're 60, 70 years old and you get Parkinson's because you had a slightly less active FOXA2 enhancer during the formation of your brain.
Right. But you might have some more of something else. These
Yes,
things
it's
act in a
all a
balanced
balance
way.
game.
Right.
Absolutely.
Right.
Absolutely. So maybe the last thing I want to show you, and we can either I'll disperse or look at more data, is one more example from Nemo Analytics. So this is now spatial transcriptomics in the developing mouse brain. And on the left side, we have the patterning center signatures that we got from Nicola Micali's monkey paper, where he painstakingly dissected out not only regions to do the single cell RNA-seq, but then once he had the data, dissected out these regional organizers in the macaque brain. So one that he defined as TTR or choroid plexus related. So this is a packet of genes. When we take this packet of genes and look at its aggregate expression in the developing mouse, you literally see the choroid plexus, either the lateral ventricle or here back, and I guess what's the fourth ventricle, is where the choroid is. So this might be sort of the easiest anatomical representation of how we can project gene signatures into data. But with the wealth of data that's out there, we also have to realize how versatile this is. So if we go up now to a study that Ron also just mentioned from Soraya Scuderi in Flora's lab, who was, I don't know if you've stayed on. Yes, Soraya, you're still here. So this is where the choroid signature shows up in your day 64 data. Now, this is from organoids that have been grown in a chamber where each organoid has exposure to a different gradients of sonic, hedgehog, and wnt. So different cell populations in each of those organoids is seeing different morphogen signals and therefore producing different cell types because they think they're in a different place in the brain. But this signature that Nicola got out of the monkey developing brain shows up here, and what they did annotate correctly as choroid plexus, and it's also wrapped up in what they were calling medialized progenitors. And this same data, of course, can be separated into where these cells were in their chamber. So just one last thing. This is the chamber. We can just zoom in here for a second. This is the chamber. Whoops. I didn't do that so well. Apologies. Well, I'll spare you the details of the chamber, but the chamber exposes the different organoids to different levels of a Wnt agonist and sonic hedgehog that's ventralizing. And so the Ds are for dorsal, the Vs are for ventral. That's how far it is away from the sonic hedgehog source. And 1 to 5 is the position. Proximal is 1 to the KIR, Wnt agonist, and 5 is away from that Wnt agonist. So now you can see the emergence of the choroid plexus signature differs by position in that chamber. You want to say something here, Soraya? I saw you jumped in.
No, you explained perfectly. Very nice. Yeah.
So, and this is just different. All those different views that we just had of your data, whether it was the UMAP or this, it's all the same data, just visualized differently here in NEMO with a different graph showing the positions. And then, of course, the key element is we've brought in gene signatures that we know and understand from other elements. So this system, in our own minds and interactions, are as versatile as we can explore the world of experimental biology going now. And whether it's NEMO or another system, I'd really like to encourage us all to interact not only intellectually, but using data in real time as we discuss things so we can bring concepts to data across the board of all these interesting studies that Ron was talking about through his talk. Because the amount of data out there that speaks to these really fundamental questions in how we vary and how it impacts our biology, it's really just exploding now. And to sort of keep it all available to us to process and use going forward is quite a challenge. So I hope we can continue to work together to do this. And if you want to learn more about this system, reach out. We'll definitely be integrating it more into our future and ongoing sessions. And Soraya's paper, what was it? Just this past week came out. Is that correct?
Yeah, the first.
So so definitely check that out. And this tool is also great for that. If you click here and any data set, you can go directly to the publication or the original data to explore it more. You can also directly download it from here. So we know we're well over our time, but I want to thank Ron for giving us what I feel is a great sort of perspective on a field that now we've been having talks for over a year on. And I'd like to encourage all of us to keep this going by thinking, by generating data, looking at data together, and thinking about who else we might want to hear from in this series to keep our ideas moving forward. Thanks again, everybody. Anyway, I'm very open to questions, thoughts, if you want to look at things or just comments on what all this means to you personally.
Thank you so much for this wonderful review. See you next time.
Okay, Soraya, thanks.
Bye-bye, everyone.
Thank you.